Thank you everybody for being here. We're very happy to have you for our webinar series for the Advanced Apple Production and Management series that we have this year. We're, I think, in our second one for the Apple sessions that we have, and we're very happy to welcome our speaker. But first, I'll introduce myself very quickly. I'm Christelle Guédot. I'm the fruit crop entomologist and extension specialist here at UW-Madison. And we're very happy to put this webinar series together with my colleagues here, Leslie Holland, Amaya Tucha, and Josie Russo that are at UW-Madison, and our colleague Annie Claude at University of Minnesota, who's also an organizer of this series. What I will do now is introduce our speaker. So we're very, very pleased to have today Dr. Christopher Adams, who's an assistant professor and extension specialist of tree fruit entomology at Oregon State University, where he's responsible for insect pest management in pear and sweet cherry. He has 10 years experience at Michigan State University in pheromone mating disruption, insect behavior, chemical ecology, and insect trapping. And we're very happy to have him today. I will just, before I pass it on to you, Chris, I will stop sharing my screen. You can go ahead and share yours. But what I would say is that we ask everybody to please mute yourselves so that we can have our speaker do their presentation, not have any kind of interruptions during the presentation. If you have any questions at all during the presentation, please go ahead and type them in the chat and we'll be happy to entertain them towards the end of our seminar. This webinar is being recorded, so I hope everybody knows that. And then I guess I'll go ahead and turn it over to Chris.
Thanks, Christelle. I appreciate being invited to speak in your series. We just organized a Codling Moth Summit last week out here in the Pacific Northwest. So I was kind of thinking about topics in codling moth management, although I didn't have a really long talk on pheromone mating disruption. So I added some detail and took some time to slow down and add some more detail. So I apologize in advance if the transitions aren't as polished as I'd like them to be. But yeah, today I just want to talk about pheromone mating disruption, and I'll use a couple of key insects as model systems and go through some of the mechanics of how we think it works. So before I get started, I am, like Christelle said, Christopher Adams. I'm at McCarrick, which is the Mid-Columbia Agriculture Research and Extension Station out in Hood River. We're a couple hours north of Corvallis, located in the lower Hood River Valley. There's a picture of our little region where fruit is grown. My responsibility covers 5 counties, 2 in Washington and 3 in Oregon. As we're right there on the river, we kind of straddle the Mid-Columbia region. We straddle the 2 states. Hood River and Wasco counties are the 2 really big fruit producing regions up here. We have, the 2 regions are very different, and so sweet cherries are grown mainly in Wasco County, and the pear acreages, most of the pear acreages are located in Hood River. And those are our 2 main crops, and the climate out here is very suitable for those two trees. So before I get started, I just wanted to point out and thank some key researchers that came before us. This is not an exhaustive list, obviously, but there's a number of important individuals whose research has really laid the groundwork for how we understand pheromones in insects and how we use it for mating disruption. So Wendell Roelofs is a professor emeritus at Cornell University and one of the pioneers in insect pheromone research. Reen Carday, distinguished professor from University of California, Riverside. Tom Baker, as a distinguished professor emeritus at Penn State. I'll share some of Gary Judd's research. He's from Summerland Research Center in Canada. Jim Miller, my major professor at Michigan State University, now retired, has spent a lot of time working on chemical ecology and insect behavior. And Don Thompson, he's with Pacific Biocontrol, and he's really responsible for developing mating disruption for commercial use, working in collaboration, close collaboration with all of these scientists and many more. Don has really been the kind of the tip of the spear on getting pheromone adopted and implementing it in apple production. So like I said, I started at Michigan State University, and my background is working with Jim Miller and Larry Goot, working in, these are this little picture of the white squares is large cages that covered a dozen apple trees. When I started with Jim, I was just an undergrad, and I started working on this crazy project where we built these huge cages. And our goal was really to just try to understand what does catching a monarch trap mean. And through that research and some other investigators in these two labs, the Miller lab and the Goot lab, we started to kind of unpack these dose response curves, and which I'll talk about in more detail, and divide up competitive versus non-competitive attraction as a couple of key mechanisms. Whoops. So I skipped ahead too many slides. There you go. Sorry. So Jim Miller and Larry Goot, pictured here, were really instrumental in kind of classifying all of the different behavioral responses that insects can have to the various pheromones that are they produce themselves and that we've come to synthesize and use to interrupt their ability to mate. And this tree, I won't get into the weeds and talk about all of these mechanisms, but understanding the specific mechanisms at play for your particular insect is really key to understanding how it works and maximizing the application of the commercial pheromones. So I'm going to focus today on just these two major classifications from that larger, more confusing chart: competitive attraction and non-competitive attraction. And I'll try to explain these two curves, these lines, and what it means and why it matters. And I'm going to use codling moth and oriental fruit moth as two poster childs for the two major differences. The one on the left is codling moth, and the graph shows catch on the y-axis over a number of dispensers. And so as we increase the dispensers, you get this curvilinear relationship with an asymptotic approach. And then the oriental fruit moth, which I'm going to talk about here in a minute, is you get this straight line relationship that crashes into the x-axis at some point, and you can completely stop males from finding females. And so those are the two major categories I want to talk about. So codling moth mating disruption has been around for more than 30 years, and we've learned a lot about how it works. It really, the other major species that was predated codling moth pheromone in the early 80s was oriental fruit moth. And from the beginning, oriental fruit moth mating disruption worked really, really well, 10 out of 10. But codling moth mating disruption has always just been good. So there's a physiological response that insects have when they perceive pheromones. So insect antennae have these tiny hairs, the sensilla on the antenna, and they are connected to olfactory neurons that extend and that extend into the sensillary dendrite. That sensillary dendrite extends into the syncytial nymph, the lymph of the antenna. Pheromone crosses that membrane, contacts a pheromone binding protein, which then transfers it down to the cell, and there's a cascade of events, sodium channels that open, and this mechanism we believed could be overloaded. So some of the early research was just on, how to provide so much pheromone that you could potentially short circuit this system. So the behavioral response, this classic behavioral response, I'm sure most of you are familiar with, is when an insect is exposed to very small amounts of pheromone, as the female might produce, small amounts, 5 micrograms of pheromone in these disconnected series of pulses at low doses, elicits a response where the male flies up the pheromone plume upwind to the source. And so this is a classic behavioral studies that have led the way on how we understand how this communication works. And for OFM, mating disruption has worked really well. So one of the early studies here by Tom Baker and Wendell Roloffs illustrated that the males have this upwind flight, but only to these small intermediate concentrations of pheromone. And when they produced high loads of pheromone, very high concentrations, that at some point the OFM stops flying upwind. Its ability to perceive and behave appropriately is overloaded. And so this study here, this is a second paper where they studied the percent of insects that stop short of the pheromone source. To do this study, they put 2 different pheromone loads, a 10 microgram and 1,000 microgram, at the center of a field experiment, released insects some distance away, and they showed that they had very high percent activation, high 80%, and then about half of them at the high load stopped at about a meter away, and 100% of them never made it to the source of the high load of pheromone. So this graph on the right is the graphical representation of that behavior. So as we add dispensers to the orchard, and if they're releasing high loads of pheromone from some of these commercial dispensers, OFM gets activated, flies towards them, but at some point there's a sensory overload and they just stop. They stop searching, and they cannot find females, and they don't complete their journey to the actual dispenser itself. And so that's why we see that it works really well. There's a specific amount of dispensers that you put in there and you get complete shutdown, which is what we want. And what we'd love to have happen in all pheromone mating disruption systems. But for codling moth, which we've been working with for not quite as long, but a long time, 3 decades now, it's always just been okay. And so this is Peter Viskel. He's a Swedish researcher, and he did some field observations where he showed that when you expose codling moth to its pheromone, it turns on its search engine. So unlike Oriental fruit moth, where the search stops when it reaches a certain threshold, codling moth just keeps on searching and actually gets more excited when it thinks it's close to this pheromone source. So it just keeps going and going and going, and it never, no matter how much we expose it to, it never seems to break that chemical perception and that in the succillum that I showed earlier. So study, so the behavioral response and the attraction of male codling moths to their pheromones was done in a wind tunnel, and Gary Judd out of Canada pre-exposed cottonmouth moth to high rates of pheromone. We have these pheromone ties, and he wanted to know if we could put more pheromone in these dispensers, can we overload the system? So to do that, he put codling moth in a jar with these pheromone dispensers for 30 minutes. And so they're being exposed to really high doses, 35 nanograms. And then he took them out and he did some electroantennogram, and he also did some wind tunnel work. And he showed that codling moth still responds and flies upwind and goes to that source of pheromone. So in these 30 years, some new technologies have emerged, specifically this aerosol emitter. And aerosol emitters are different than the passive pheromone dispensers in a number of ways. One is just the rate of pheromone. So on this chart, I'm showing, the female produces 5 nanograms. These hand-applied dispensers are at 5,000 nanograms per hour release rate, and an aerosol emitter is just huge amounts of pheromone. But even at these high rates, we find that codling moth still is responsive. So this is a study done by Pete McGee to find out just if we could, just how much pheromone can they take. So to do this, he took an aerosol emitter and he sprayed the limb of an apple tree. And then he caged codling moth. He bagged that limb and then released moths into there and held them there for 24 hours. He did, I think, a couple different doses here, 5 sprays, and then a lower rate of a single spray. And then after 24 hours, you release them in a no pheromone mated disruption orchard. And this data shows that the aerosol emitter exposed moths became super searchers. They didn't get shut down. We actually caught more in those environments. He also did it with the the passive dispensers, the hand applied little ropes, where he put a number of dispensers in the bag on that limb and held them there for 24 hours and then released them into an orchard with pheromone mating disruption in the yellow, and then a block with no pheromone mating disruption. And again, you see that they're not shut down, and we actually catch more when they're pre-exposed to those high levels of pheromone. And so this is why we get this curvilinear relationship on codling moth. So as we increase the number of dispensers on the graph on the left on the x-axis, we get an immediate reduction in catch, but then that return on investment is really an asymptotic approach. So there's an inflection point and we just don't get a return on investment. And no matter how many more pheromone dispensers you add, no matter how high of a volume you add to the orchard, codling moth just keeps on searching and finds a monitoring trap and or females, which is why control is so difficult. So I'm going to kind of be nitpicky, and this is a classic diagram that we see all the time in the Pacific Northwest Handbook and lots of extension bulletins of a male finding a female moth and then a pheromone mating disruption. I've kind of got 2 kids now, and I spend a lot of time at the doctor's office with Highlights magazine. So I'm kind of poking fun at this cartoon. And it's really just a cartoon to help visualize what's going on. But I think it's really important how we think about this system really influences how we use it or try to use it. And so understanding exactly what's going on is important, and misconceptions, or however subtle, really impact how crop consultants advised farmers on how to apply it and how research is funded to, dig deeper and to advance other insects, pheromone mating disrupting technologies. So how does it work for codling moth? It's competitive disruption. There's questions about, and research has been done about where we hang dispensers and how many we need and how much pheromone is in the dispenser. And a lot of that's been worked out. So as we think about codling moth and how this technology is applied, we can really be critical of this cartoon and nitpicky, yes, but it's important. There are important criticisms. So number one, the volume of pheromone pictured is probably inaccurate. These are really big. It looks like the tree's on fire. That's really not the amount of pheromone that's coming off of those dispensers, not the commercial dispensers. There's multiple dispensers in this tree. That's also not how we apply it. The dispensers are hung low in the tree. We know that codling moth in particular and needs to be hung at the top third of the tree. So you would never have dispensers down there. Because the plumes are so big, it looks like in this cartoon, they're all overlapping, suggesting that the male can't find the edges of any of these individual pheromone plumes and get find its way to the source. And from this cartoon, that jagged line, it looks like he never finds a female. So this just isn't how codling moth mating disruption works in the field. So we probably shouldn't have those cartoons in extension bulletins because it's confusing. Instead, this is how codling moth pheromone mating disruption really works. It's a numbers game. So even with really high levels of pheromone loaded into these lures, the male codling moth makes his way upwind and finds that lure. When he doesn't find a female, he keeps going and he finds the next one and the next one and the next one. And eventually, given enough time, he finds a female and we have mating and then oviposition and fruit injury. But it's because it's a numbers game. If we can even delay mating just a few days, the lifespan of the adult codling moth is only a week or so. So this system works okay. And if we have the right number of dispensers in the field, because the window of opportunity to mate is really small and the female's fecundity goes down very rapidly too. So we only need to delay this mating 3 or 4 days to really be effective. So how many do we need per acre? So here's some research from Jay Bruner of Washington State University. And this on the left, just reminding you, this is that curvilinear relationship that we see. So catches on the y-axis, and on the x, we've increased the amount of, this is a codling moth twin tube, CTT, stands for twin tube. And these are loaded with pheromone and put out in the orchard at 50 per acre, 100 per acre, 200 and 400 per acre. And the graph on the left in blue, that's with high populations of codling moth. And you see that we get this concave curve. And at some point, if we were to add, the data isn't here, but we've done some of this research, 800 or 1,000, you would not continue to see that catch go down. The blue bar would stop progressing towards zero. By contrast, if you have low populations, so if you have really good codling moth control, if you're part of an area-wide program, you still have that curvilinear relationship, but you get a much higher impact. And so 200 is not really different than 400, and we get really good control even at 100. Of course, at low populations, every control mechanism works really well, but this just illustrates that this is a numbers game and it's density dependent. So unlike a pesticide, codling moth mating disruption relies on getting the population to low numbers. It doesn't work well when we've got huge wild population or abandoned orchards across the road. It's a really good foundation to a management program, but it can't shut down all mating. It's just it is just one of the many tools of an IPM program. And so I've got some extra graphics I forgot here. So that's that curvilinear relationship. And then at low populations, you can cut down the rate. But researchers come to this sweet spot of 400 dispensers per acre gets you the optimal return on investment. It doesn't make financial sense to add more dispensers than that per acre. So what about these aerosol emitters? So Dr. McGee also did research with aerosol emitters. And again, we get this same curvilinear relationship where as we add, as there's one dispenser every 4 acres is in red, one dispenser every 2 acres in green, a dispenser every acre, et cetera, et cetera. And what's notable is the red line is an equivalent amount of disruption to the maximum rate of 400 per acre of hand applied dispensers. I think what's notable is that even at 6 dispensers per acre, catch is still not 0. So that's really important to keep that in mind, that this is a foundational base of an IPM program, but it's not a silver bullet. And for codling moth anyways, we're never going to get to complete shutdown. So it's really important to also think about timing of sprays and larvicides and a full management program. And again, these rates are really based on that they're all relative to high populations. So these data represent relatively high populations. If you can get the population low, you can get better control and use lower dispensers per acre. The recommended rate from the manufacturers is 1 per acre, but you can imagine if you can lower that blue bar and all of those catch numbers are going to go down in a relative fashion, that you could use fewer dispensers per acre. One of the problems with aerosol emitters is that they do a really good job of creating big plumes and shutting down all of the traps downwind of the emitter. So here is data from Walnuts in California, where they have traps in a grid. The trap spacing is pretty wide. You can see that x-axis is almost 500 feet in between those on the downwind side. And there's an aerosol emitter or a puffer is one of the terms used by one of the companies. So the P is a pheromone puffer in the center there. And you can see the data, this artificially created heat map is zeros are in purple and low numbers are in blue. And what happens is all of your monitoring traps get shut down. So it looks like you have really impacted the male moths in the orchard. But it's important to remember that pheromones are not pesticides. And so no trap and a catch doesn't mean that they're dead. What they do is they follow that big plume up to its source, and then they get behind it into clean air. And then they can find those monitoring traps on the upwind side of it. And again, these data are not real. This is an artificial heat map. There's no data points in between the 80 catch in that 800 meters away from the 0 at the aerosol emitter. And what that means is, you don't have to be meters, hundreds of meters away from the aerosol emitter. We have shown that if you just put a trap 1 row upwind of that aerosol emitter, you will catch codling moths because they've followed that plume all the way up. They get into clean air where the aerosol emitter is not blanketing the low load pheromone lure in the monitoring trap, and you get catch. So this is really critical to how you crop consultants place traps and it's important to think about, where, if you want to know whether or not you have pressure, you should still have some traps down there, because zeros means that's real data and it means that your management program is working, but it doesn't mean that you don't have any moths in there trying to mate. And so you want to really focus on edges, especially on the upwind side, and you may want to increase the number of traps on the leading edge of that block. So I talked faster than I thought. I'm down to my summary slides. So in summary, I just want to make sure we hammer home these take-home messages. Oriental fruit moth works by non-competitive attraction. So you can shut down Oriental fruit moth using mating disruption and really break the cycle of mating. Unfortunately for codling moth, the mechanism is competitive attraction. So it's really a numbers game. And understanding the physiological and the behavioral response of insects to their pheromone is really key to optimizing mating disruption for these particular species and any new species like San Jose scale and these other insects where we're investigating the possibility of using this as a tool. So the benefits of mating disruption and even for codling moth where it doesn't, it's just good and not great, is that even good mating disruption for codling moth can shut down damage by 80 to 90%. So it's a really important tool for an IPM program. And that's because delaying mating just a few days can drastically reduce the female's fecundity, the amount of eggs she can successfully lay, and the success of the males in finding her. Mating disruption is a selective control, which is really important for us out here where we're growing a lot of pears. And pear scilla is our key pest of this crop, and we rely very heavily on natural enemies. So this is a selective tool and allows natural enemies to remain in the system and build their numbers. So it's a targeted non-pesticide control and works really well in this system because codling moth is also a pest in pear. So for recommendations, mating disruption should be the foundation of an IPM program that includes trapping and scouting for damage. It needs to be applied, it should be applied early before leaf out at a rate of 400 dispensers per acre. It needs to be hung high in the trees. If you're going to utilize the aerosol emitters, they are an effective tool, but it's still a numbers game. And so you really want to make sure that your population is low. An area-wide system works best. So if you and all your neighbors are doing the same thing, that's the best approach because an unmanaged orchard across the road allows mating to happen and then those females fly over and that patchwork quilt of management programs creates large problems for the industry. So in closing, I just want to thank, there's a large staff here at McCarrick that is working closely together, including a brand new person at USDA ARS, Rachel Lesso, and Kelsey Galimba is our new horticulturalist. And I really want to thank Jim Miller and Larry Goot, who are my mentors at Michigan State University. We just recently lost Larry Goot to stomach cancer. So just wanted to acknowledge him, and that's his son, Tommy. Tommy's all grown up, but Larry loved pheromone mating disruption, and he really loved being part of this development of this new tool that get us away from pesticides and get us back to a kind of an IPM. And then Don Thompson was generous and shared a lot of the slides that I showed today, and Pete McGee, who worked with Larry for 17 years before transitioning into Pacific Biocontrol. He's now in charge of research and marketing over there. So sorry, Christelle, I talked faster than I thought. I thought I added enough slides to stretch this out. I'd love to entertain any questions that you have from the group.
Yeah, well, thank you so much, Chris. That was great. That was very informative and a lot of basic information, mating disruption that I think people really need to understand in order to think about how we implement mating disruption. There's one question in the chat right now asking about product names for mating disruption. Do you have any names that you can provide? And I mean, there's several companies, and of course, I'm sure you don't want to endorse any company over others, but there are several companies.
There are several companies, and you're right. I don't want to necessarily endorse anyone. Pacific Biocontrol is one of the first in the game. There's Sutera and Trace and Semios, which all make aerosol emitters and hand applied. And I think that they all have the same, a very similar formulation. And we at MSU did some research and were part of the R&D as they rolled out those new products. And they all work in a very similar way. I would caution you that if you're a grower and some of these products are labeled with as only needing to be having twice the amount of pheromones so that you can use half the rate. And I would argue that we have research to show that that claim is not correct. You can, if your numbers are low for codling moth, if you can control the population with conventional pesticides or through an area-wide program, and you and all your neighbors are timing your sprays really well, and you have low numbers, you can use half the rate. But it's not because you doubled up the load in the lures. I hope that came across in the talk, that you can't overload codling moth with pheromones. So selling a product with twice the rate in each individual dispensers and suggesting it can be dispensed at half the rate per acre is the wrong that's an inaccurate way of describing the system. So be careful when some of those labels are not vetted by research.
Well, thank you for mentioning that, Chris. I guess one question that I've heard from people is, because like you said, you don't disrupt 100%, you always have those low numbers that end up in your monitoring trap. And I want to make sure people know that it's very important to have monitoring traps and check that this is working and to determine when to spray. And so do you have any recommendation of when you're trapping some in your monitoring trap, what's a threshold number you would use to figure, okay, now I have too many moths in my orchard, like it's failing for some reason or another, I need to spray? And I know there's a lot of conversations on that and not an easy answer, but if you can elaborate on that a little bit, I think that would be helpful.
Yeah, no, we do have action thresholds. So Jay Bruner in the Pacific Northwest established a threshold of 2 to 3 moths in a week, on 2 consecutive weeks, indicates that you are going to get economic injury and you should take some sort of chemical, you should apply some chemical treatment to get those populations under control. Larry Goot in the Midwest had the number a little bit higher, 3 or 4 per week for a couple weeks in a row. There are some really good models.
I think you're talking about outside of mating disruption, right? Like in general, the population. I was asking about when you have mating disruption and you're catching some moths. Is that what you're referring to?
Yeah. So I think the action threshold. So with some caveats, with a little background, without mating disruption, the monitoring trap, the way we used it initially was a really low load, what's called an L2 pheromone dispenser in a trap. If you have pheromone mating disruption, passive dispensers, hand applied dispensers, they would compete with the trap and the female and probably shut that down. To address that, in mating disruption orchards, there's a 10x lure, and it is 10 times the amount of pheromone. And so it catches really well in those mating disruption orchards. And so in that circumstance, the action threshold is the same, because you're still getting numbers. What you're tracking is the beginning of that first generation of the phenology. And that lets you know that you are, adults have emerged and started mating and likely laying eggs. And so that's a time to target an ovicide or an oil, because you're going to see early stings. There's a ton of other lures out there, which has complicated the system. We don't have good research to back up. Alan Knight has been key in developing lots of plant-based volatiles. Pear ester has been shown to be really attractive to males and females, along with a number of other plant volatiles. Alan Knight, it feels like he comes out with a new lure every couple of years, which is great. It's great to see the science advance, but I don't have action thresholds to match all of the new lures, because if they're catching more of the same population, it doesn't, you probably need a new action threshold. But the really the key is to time your sprays for that first generation egg laying and those first hatches, and understanding the degree day models, when you can expect those, using monitoring traps to confirm the models and to give you a sense of your relative population and whether you have high pressure or low pressure in your orchard. So you can use, and I'm not going to say not to use those really effective lures, but I don't have an action threshold that transfers. If you're catching, I think Alan has suggested that some of them are catching 10 and 20 times as well. I just can't speak to how that impacts your action threshold. And a lot of those decisions are really based on the grower's tolerance for damage, whether they have a high value, product, or if they're going for juice or cider. All of these things need to be discussed with a crop consultant and the grower to decide when you need to take action.
Thank you. While people keep putting questions in the chat, I'm going to launch a poll that we have about this webinar. So if you could please answer the poll, and we'll continue discussing while everybody is filling out that poll. I hope you can all see it and are able to click on it. I had one comment and then a question. So are there any questions that people have that they would like to unmute themselves and ask live to Chris?
I'm not aware of of Wisconsin's apple acreage, which is embarrassing since I spent so much time in Michigan. But are there large contiguous acres of apples?
It's a little bit different. In Wisconsin, we have apple production, but it's pretty much spread out throughout the state. Yeah. And we have, it varies in size of the orchards. We have some large orchards and we have some smaller orchards. I would say, I don't know what the total acreage is off the top of my head, but on average, we're going to have orchards that are less than 10 acres.
That's one of the weaknesses of mating disruption is it really works best on large continuous acreage, which you probably know, Christelle, from being out in the Pacific Northwest, is that you were there during that time when Jay was kind of trumpeting this technology. Works better when everyone in the area is doing the same thing, and it works better on large acreages. And that's always been the challenge in the Midwest, unlike the Pacific Northwest, is there's so many wild apples out in the woods that are moved around by deer, and there's crab apple, and there's all of these ornamental trees in backyards and on golf courses, and it really becomes difficult to control everything. There's a lot less of that patchwork quilt in the Pacific Northwest, and so getting an area-wide program organized was a lot easier. But it can be done. Larry spent a lot of time when he moved out to Michigan 20 plus years ago, just meeting with growers and telling them how this technology can save them on sprays, and getting them to try it, and then convincing their neighbors to try it. And so it can be done. And I think it's an important tool in any IPM program.
Yeah, and I think people, I don't know if everybody that's online right now is from Wisconsin. There are probably people from Minnesota and some other states around, but I think people in Wisconsin are pretty well aware of that if they have smaller orchards, that's going to be more of a challenge. And one comment that I wanted to make is that Chris and I are on a group of people that are really trying to have large discussion about mating disruption and what is the future of mating disruption and how can we help people. So hopefully we have a pre-proposal for a planning grant just to discuss and figure out how we can, what are the, the benefits and disadvantages of mating disruption, and how can we best implement that in the different systems we have across the nation. So hopefully that will be something that will be able to update people on. There's a question in the chat from Sarah. My winds are dominantly south and southwest. Which row do you recommend to start the mating disruption lures if you are facing the first row?
So if is she talking, I assume she's talking about aerosol emitters. So the first thing I would do is I really am a fan of the hand applied. If you just have a few acres, you can imagine if you have 500 or 1,000 acres, that the labor involved in hanging hand applied can be prohibitive. And so the aerosol emitters is a really good tool to address the time and the difficulty with getting labor in early in the season before leaf out. But if you have a small number of acres, I am really a fan of, and because of that curvilinear relationship, because it's a numbers game, of hand applied. And so if you're going to do hand applied, the wind direction is not as important. Your monitoring traps should be focused on historical hotspots where you've had injury. You should focus there and react to the earliest amount of the earliest timing of trap catch and spray the whole block based on that hotspot. If she's talking about aerosol emitters, then you need to have that trap on the upwind side and be aware that you have not stopped male moths from finding females. You've stopped them on the downwind side, but if they're mating outside the orchard or on the borders on the edge of the orchard, you can have problems. So if you're trying to address a labor issue and hand applied isn't an option, one hybrid solution is aerosol emitters on the interior and then hand applied on the borders where you might expect some immigration or some vulnerability to keep just kind of a blend of the two technologies. It's once you've done an entire block with aerosol emitters by yourself, instead of bringing in a crew of workers, it's really enticing to keep doing it that way. But I just caution you because it's easy to get false negatives in monitoring traps that are incorrectly placed, and it really doesn't it makes it more difficult to trap at all, and doesn't, it's hard to know what's really going on. And then that's one of the reasons, I think, that one of the many reasons why we had to form a codlingmouth task force here in the Pacific Northwest that I'm heading up, is because of a shift to organic pest management. That's one, because of the draw of the cost, where the market value of organically grown fruit has increased. So lots of people are trying it. And I think a lot of people have been lured into using aerosol emitters, which again can work under low pressure, but they make it difficult for you to know that you're no longer under low pressure and that numbers have built over the last couple of seasons. And then you find yourself in trouble, and we no longer have glutathione and some of these broad spectrums to get you out of trouble. And so to get out of trouble or high pressure, you now need to do everything right. You need to time all of your oils and your larvicides perfectly. You need to monitor more. You need to, get back to hand applied mating disruption or protecting the borders. You need to talk to your neighbors. Anyways, so I know that might not have been exactly what her question was, but it's there's a lot to unpack in how we use this. But even without area-wide mating disruption, I really think utilizing mating disruption, even on a small block, is important because we don't have any new chemistries coming down the pipe. So it's 10 years of R&D to bring a new pesticide to market, and there's just nothing in the pipeline. So we're looking at least a decade, if somebody starts now, before we're going to see any new chemistries. And the more we use these less effective chemistries that have a smaller residual time, and so we end up using them more frequently. We just accelerate resistance, and we're going to get into trouble. So I would really encourage everyone to use IPM as the basis for their management program and then build from there.
Thanks, Chris. We have a couple more questions, but before I do that, I want to share with you the different websites where you can go back and look at the recording of this presentation. If there's anything you want to revisit, we put the recordings on those different websites. You can access them there, and you can just go and listen to either Chris's talk or even other webinars that will place there during our webinar series, whether it's grape, apple, berries, all of those will be present in those different websites. And also I put on the shared screen our next webinars that will happen on March 24th for bloom thinning, and that will be Maya Tucha, and April 14th on ground management. I think that's Annie Claude that will be presenting on that. I just wanted to say that and then go back to a couple questions that came.
Yeah,
so
I was looking.
were you looking? Okay.
She's clarifying her question, and somebody else helped. So I don't know if I talked about monitoring traps. We have shown over time that the best gauge of your pressure is at one monitoring trap every 2.5 acres. There's a question about CMDA pheromone lures. The DA is a pair ester, and in the Pacific Northwest, it seems to catch males and females, which is great. I have personally done some research in Michigan, and for whatever reason, I don't know if it's the background plant volatiles or the humidity, we don't catch, I believe the data in Washington, and I've seen it and done some of it myself. It does catch about 50-50 male and female, but it doesn't seem to work in the Midwest. And I don't know why. I need to partner with a chemist to see if we can capture just orchard volatiles and see if there's a reason. But where I was going with this is if you're catching males and females, you now need to sex the moths because your action threshold should be half if you're going to stick with that 2 or 3 moths, assuming you're catching males and females. Do you see how these are all areas where we need further research. Every time we change the lure and change the way it catches or add, we start catching females and males, all of those action thresholds are based on male only in non-mating disruption orchards or the 10x lure in a mating disruption orchard. And so that it just gets more complicated. Tim Spencer was trying to clarify, if you have 100 rows, do you place the monitoring trap in row 2 through 3 so you have a check monitoring? So that's where you want one every 2.5 acres to really have a sense of what your pressure is. You can do fewer, and out here where crop consultants manage 1,000 acres, they do fewer, but you have to know your orchard to know if you're only putting one every 10 acres and you've got hot spots, you're missing them. And so you're taking a chance. If you know your orchard and you know the whole interior is usually pest free and all of your damage usually happens in that southwest corner, so that's where you concentrate most of your monitoring traps just for your time management, that strategy makes sense. But if you're not monitoring, then you don't know.
Yeah, and knowing your hotspot, I think, is very important, and you're completely right on that. People have to really pay attention to where you have those hotspots, where those mated females are coming in, because that's the thing. The females are still there, and so if they are mated outside and come into your orchard, mating disruption falls apart. I mean, that's why it doesn't work in small orchards, and I think people know that, but it's still a good reminder to remember that that's important to pay attention to.
Danielle asked to follow up, would I use only the 10x lures in an MD orchard? I would use both. I wouldn't rely on just one lure. The lower load lets you know that your mating disruption strategy is working because it's much closer to a female release rate. And so it shows you your mating disruptions working. So I would mix it up. I'd use a few. I'd use both. How far will a laying female fly? So there's been some research on this, and it depends on her mating status. So virgin females fly a little bit further than mated. Once they're mated, they don't either behaviorally or physiologically just don't have the drive to go as far. If they're near apples, they'll start mating. So quarter mile on the high side for females. Probably more realistically, it's just a few hundred meters.
Yeah, and like you said, if there's apples around, is there an incentive to go somewhere else once you're?
Yeah, we've found over time that hotspots tend to stay hotspots. So a lot of growers and consultants kind of know where they are always getting damage. And so focus on monitoring in those places where, you know, the larva, last year's larva overwinter very close to the tree that they came from. So you can kind of predict where your hotspots are going to be year over year. Good questions.
I guess you answered, I shouldn't say we. I guess you answered all the questions in the chat. So if people have any more questions, please continue to put them in. If not, we'll thank everybody for joining us, and very much Chris for spending the time and talking to us about mating disruption. We're very happy to hear about all the work you've been doing, and then of course we'll follow up with you once you have a bit more your feet under you at Oregon State University and doing a lot more research there, to give us an update on what you're finding. And yeah, pairing up with a chemist that could look at the background odors.
Yeah,
that'd be
something you and I could work on. We really need to understand why the female lure doesn't seem to work as well in the Midwest. And then we here, the Cottonmouth Task Force will be posting a bunch of talks. We did a day of talks on this same topic, on a whole host of Cottonmouth topics. And I think that Gwen Hoheisel at WSU is going to make those recorded talks available if anyone's interested. They're not up yet. We're looking for a platform to post them, but those will be coming out soon. And I'll send you a link so that you can pass it on to your stakeholders. For interested people, they can go get those talks.
Yeah, that would be great. I think there's a lot of interest for mating disruption, but a lot of, I wouldn't say resistance or reluctance. It's not that. I think people are interested. They're just not sure that they're going to get, you know, the kind of control that they're hoping to get. And so there's more and more people that are coming on board with mating disruption, but there are trade-offs, of course, and people recognize that and want to be cautious before they implement that in some of their, you know, possibly smaller orchards as well. So there's a lot of work we need to do on that, on those smaller.
Cottonmouth is just one of many pests, so you have to think about your return on investment, because you still have OFM, you still have there's lots of other things you're doing.
Yeah.
But yeah, one piece of the puzzle.
Exactly. Well,
thanks for inviting me. It was sorry I didn't get to see all of your guests to know who attended, but hopefully it was informative, and I'm looking forward to our collaboration on further pheromone research.
That would be great. Thank you so much, Chris. Thank you, everybody.
Bye, everyone.
Thank you for your nice comment.
Thanks for the great questions.
Thank you. Bye-bye.
Bye.